is just sort of how well connected, summed over all of the nodes within a segment, how well connected those points are. So the problem is if you have two segments and one is very small, it's going to have a very small value. And if that denominator becomes a small value, that whole element becomes very large. So even though the other element might be a small size, the fact that this one became small will penalize that a lot. So by doing this normalization, you can end up with a much better cut of the graph. If you just wanted to do the best end cut, normalized cut, that would be computationally prohibitive. But there is an approximate solution that exists using what's referred to as the generalized, or solving the generalized eigenvalue problem. I'll just show you a very simple, not simple, I'll show you a very high-level description of what goes on. And if you want more details, you can go take a look at papers or Wikipedia or anything like that. And so here's how it works. Let's suppose we've got some matrix W, which is the adjacency matrix. So Wij is just the adjacency between pixel I and pixel j. And then there's a special matrix, this diagonal matrix, that is just what's called the degree of the node. For every vertex, how many other vertexes are connected to it, right? And so in this case, it's the sum, so it's a diagonal matrix, so the degree of pixel I is just the sum of all the affinities ij summed over all the j. And then what you can show is that the normalized cut cost can be written in using this simple expression, where this vector y is what's referred to as an indicator vector. So it's a vector that's got positive ones and negative ones as to whether, remember, we're cutting this into two parts, A and B, so it's a positive one if that point belongs to segment A, and it's a negative constant otherwise. All right. So to solve even that for y is very difficult, but what you can do instead is, this is just written out here, we solve this eigenvector equation, and what that'll do is it'll solve for you for these y's directly. But these y's, they're not going to be binary, you know, -1 or +1. They're going to have continuous values, and what you then do is you use that vector in order to solve for the partitioning. And this is related to spectral partitioning, for those of you familiar with those algorithms. And it's just an eigenvalue way of approximating the solution to this partitioning, because the full solution would be computationally too difficult. So let me just show you some of the results that you can get using that. All right. So here are, again, this is taken from the Berkeley database, and because Shi and Malik, Jambo Shi and Jitendra Malik did normalized cuts, and you can see that there are some pretty good results here. You know, you like, especially like this one here, where it breaks up the picture of the people in a particular way. Now, of course, these segmentation is tremendously influenced by that affinity matrix, and the affinity matrix is defined by that distance function. So we're back to feature spaces again, right? We have to know what features we should be computing a space over. We may have to scale the features, because all of that determines the distances. But once you do that and you play with that, you can end up with a really powerful segmentation method. By the way, you'll notice that there are more than two segments in these pictures. Well, so how do you do that? Well, the simplest is you partition your graph, and then what do? You partition again, and you need a stopping criteria based upon sort of the affinities of the links that you're cutting. Here are some more examples also taken from the Berkeley database for segmentation. So that's the normalized cut approach, and it's one of the more effective methods within the whole graph-based way of doing segmentation. The pros are, because it's a graph structure, we've got lots of algorithms for dealing with graph structure, so it's a very generic way of doing it, because you separate out this graph structure question from the affinity matrix computation. So the affinity matrix is where you get to play with things like your distance functions and your feature spaces, and then your graph algorithms just run given that matrix. So again, it doesn't require a model of the data distribution, it just requires a distance function that respects that. There are some challenges. In general, the complexity of dealing with this can be pretty high, because you now have these matrices that are essentially n by n, where n is the number of pixels. So you want to be careful exactly how you manage that complexity. If you have densely connected graphs, because you want to allow for large regions, etc., the system has to work even harder, and the spectral decomposition approximation to the normalized cut is a little bit trickier to do. And then there's this question about this balancing of the partitions. So you remember we introduced the normalization to the min cut in order to not get small pieces, because there was a bias towards small pieces in the algorithm. Well, another way of thinking about this is we've introduced a preference for balanced partitions, for balanced regions. So if you really wanted to have small little spots being pulled off of something that was bigger, normalized cut finds that kind of painful because it wants to have these balanced partitions. Having said that, I will say that it is one of the more effective segmentation methods that people currently use. And I think it's because it has this nice dissociation between the affinity matrix computation and the graph segmentation method. So that ends our conversation about segmentation. You can go from these sort of very simple methods that just cluster in some feature space to ones that cluster the feature space and then do histograms and cluster on those, to the more complicated graph-based methods which use some form of spectral partitioning in order to split the graph into multiple sections. Welcome back to Computer Vision. This is another lecture of mechanisms or methods that are they're not sort of current computer vision research or really the state of the art in computer vision, but they are very important methods that we make use of all the time for a variety of operations. And what we're going to talk about today is really some binary image analysis. There are lots of times that we produce images that are essentially zeros and ones. One talks where 0 is a background and 1 is a foreground. So, for example, I might be looking. In fact, we talked about background subtraction when we were doing detection of moving objects, right? And so basically, the ones were where the stuff was supposed to be moving, and the zeros were supposed to be where it wasn't. And in fact, there was this example that I showed of people sitting on the steps of the university, and we did a median filter, and then we subtracted them off. We did a thresholding. Then I said we did something called morphological operations to clean it up. Today, we're doing the morphological operations, and it's often buried deep within any functioning vision system. So, I mean, here's an example of just showing a binary image, okay, where you've got a circuit with some leads that are connecting it, and some of those leads have shown up well, and some of them have shown up well, or maybe some of these things are noise and you want to get rid of them. And here, of course, this is a notional version of a binary image where you've got ones and zeros. So binary image analysis is used in all sorts of things. We're just showing you sort of document inspection, but parts inspection. So you've got a part and it's supposed to have particular property to it, and you don't want to make sure there are any holes that shouldn't be there, or the holes that are supposed to be there are there, mostly for two-dimensional stuff. Also manufacturing. You have some idea of what the images are supposed to look like, but it's basically where is there stuff and where is there not stuff is what matters. And so you can think of those as binary images. And of course, document processing, where basically, if you think of it as just black ink on a white background, it might be very fine, but there's not really supposed to be gray. Now, those of you who know about anti-aliasing, you know that has to do with limited resolution. We're not going to talk about that. We're just going to talk about sort of binary images. You know, what kinds of operations do you want to do on binary images? So, you know, often what you want to do is you want to separate objects one from another that are sort of accidentally touching, and you want to collect together pixels that all belong to the same object. And then another thing you might want to do is compute the features of a binary object. And we actually did this also. Remember we talked about hue moments, and we were talking about moments in general, and moments are a descriptor of the shape of a binary patch. And so, again, those are things that you would do on binary images. So here's an example. This was a thresholded red blood cell image. And by the way, in the old days, not so old. In fact, my mother, I think her first job out of college was as a medical technician. One of the things you had to do was count white blood cells under a microscope, and for a certain amount, and you would count the density. Actually, people sometimes still do that, even today. But what you'd really like to be able to do is spread out a bunch of blood cells and have a machine do the counting. How many red blood cells? How many white blood cells? Especially if instead of just doing a patient at a time, you were doing a massive study and you had tens of thousands of samples you wanted counted, right? So here's an example of taking an image, thresholding it, and You know, if I were to ask you to count how many red blood cells there were, you could probably find all of the, well, let's start with one that's easy, right? You'd say, aha, here's one that's pretty separated, no problem. Here's another one. Oh, but this is a weird one. Look at that. It's got a hole in it. And here are two. Those are probably two, right? But it's one blob connected by a thin thing. So you'd really like to be able to take this image and then fix it up. You know, because in this particular image, there were 63 separate objects detected, and a normal blood cell has an area of about 50. But you'll notice there are some things here where obviously there were things that were touching each other that were counted as single cells when actually they were gobs. And then there were things that were really small, right? And so here they were just using the number to determine which kinds of things are cells. And what you'd like to do is you'd like to be able to separate out each one of those cells. So we're going to do some binary image operations to do that. And what kind of operations are there? Well, first one we're going to talk just a little bit about is thresholding, because in some sense, in order to get a binary image, you have to go from a color or grayscale image that has continuous values, or lots of values, to something that just has 2, 0, and 1s. So determining a threshold is always a challenge. Then what you'd like to do is you'd like to find all the pixels that are connected. And that's called connected components analysis. And we'll talk about that. That one's used a lot also to find the independent blobs. And then you want to sometimes improve those blobs, and that's referred to as morphology. And we're going to talk about a variety of morphological operators, including some of the more important ones. Then once you've done all that, then you may compute some features on those blobs, like I was talking about before, moments, areas, these kinds of things. Let's talk about thresholding, and this is an example. I think this might be taken also from Linda Shapiro. So the idea is that you're imaging these cherries, okay, and the idea is that the background here is very dark, okay, and a healthy cherry is kind of bright, all right, but a bruised part is medium dark. And what you'd like to do is be able to say, do I have a bunch of bruised pixels? So in this particular one, they've removed all the black things, but you see these are the grayscale values, right? And you and I look at this and we say, yep, you know what? Here's my threshold, and this is the number of pixels that are bruised, and this is the number of pixels that are good. And the idea of finding a threshold like that automatically is not trivial. Generally, what you do when you're looking for thresholds is you take a histogram of the intensities, right? So this might be, go from 0 to 255, and this is the number of pixels that you have in the image. And the question is, what are your distributions? Unlike this one, which had this nice, clean separation there, this one, it's a lot more complicated. Are there 2 or are there 3? I think, for my take, I'd say, there's 3 lobes there, right? And there's different ways of trying to do that. A simple method, this is again a long time ago, Atsu's method, it's a binary vision method that basically says assume that your system is bimodal and find the cutoff that minimizes the within group variance. Okay, so you're only allowed to draw one line. And it's weighted by essentially these values. And when you do that, what's kind of cool about it is you can do that when it's certainly bimodal. When it's actually trimodal, let's say, you'll typically pick one of the modes, all right? And then you can do it again, and you'll cut it again. And what's kind of nice is when you slice a mode that shouldn't be sliced, you'll get a reduction in the variance, but not very much. And you can track how much that is. And the method can be adapted to do a pretty good job of thresholding a continuous grayscale into either a binary or a 3-valued system. This gets done sometimes in medical imaging. So here is an old scan, probably I'm going to guess CT, and the image on the left is the grayscale, and then they've thresholded the image on the right. And what you'd like to do is you'd like to say, there's this chunk here, and there's this chunk there, and maybe this chunk and this chunk, and some things there and that. And maybe I'm going to ignore some of that other stuff. The first thing we're going to look at is what's referred to as a connected component analysis, all right? So some of these are a little hard to see, but we'll just focus on this one right here. The idea is I'd like to pull out this thing as being distinct, say, from this thing, okay? These are not connected, so I would like to label this maybe as a 2 and this is a 3, right? I want to find all of the connected components or a connected set of pixels. The connected components algorithm or operation takes a binary image and it generates what's referred to as a labeled image. So every pixel now, it used to be